Good morning. In this session, we are discussing the next kind of simple tissue, that is from the fundamental tissue, the parenchyma. The third type of simple tissue is differentiated, and they are also the same as it is a collection of similar cells with a common function. So, accordingly with the function, it is the fundamental tissue or the simple tissue is de-differentiated, and here we can see the third type of simple tissue, that is which is termed as sclerenchyma. So, what are sclerenchyma? These are the tissue consisting of dead cells, that is a group of cells differentiated, and this is a fundamental tissue which is getting de-differentiated into sclerenchyma. Okay, and in due course, according to their functions, the cells become gradually dead. How it is getting dead? Normally the cells are with a thin walled nature, which show the intercellular transport, but here this tissue, that is the sclerenchyma, required to differentiate as the as to perform the mechanical function. So we can see the sclerenchyma is usually seen as dead cells in the plant organs, and to show how or why they are become dead, because it shows the lignified or unlignified walls. So the cells are getting differentiated primarily as thin-walled parenchyma cells or originated as thin-walled parenchyma cells, and to perform a particular function, the cells become gradually deposited, that is the cell wall become gradually deposited with a more lignin, and thereby the cell do not perform the transportation, that is intercellular transportation. So here the structural differentiation, it is the cell wall is either lignified or unlignified walls is the peculiar characteristic feature of sclerenchyma, and it give hardness to plant organs. So it is the function of sclerenchyma. Why the cells become lignified and gradually become dead? It is because of it perform or it give the hardness to the plant organs. So it is the special feature of sclerenchyma, and are of two types. Here also the sclerenchyma, according to the differentiation and de-differentiation, the cells become differentiated into two types. One is which is called sclerids, and the other one is called as fibers. Okay, and the sclerids, what is the differentiation? How we can differentiate sclerids and fibers? Of course, while see the structure or internal morphology of the sclerenchyma cells and whether it is a sclerids or a fiber, it is easily we can differentiate according to their histological differentiation. So sclerids do not show any pointed tip, that is the cells do not no pointed tip, and while we observe the structure of fibers, another kind of sclerenchyma, here we can see the cells with a pointed tip, and also the fibers may show pit cavity with a narrow lumen. So that is the difference. That is the sclerenchyma. Yes, of course, the sclerenchyma is getting differentiated into two corresponding to their structural differentiation. Here in this diagram, you can see the two types of sclerenchyma. That is, one is sclerids and another one is fibers. What is the difference between the early mentioned collenchyma? Of course, the cell wall in collenchyma, it show localized thickening, but while we observe the structural differentiation of sclerenchyma than that of collenchyma, here all the walls that is inner and outer wall. Here you can see the diagram is with a middle blue lined marking, and it represents the actual thin walls of the cell. But gradually what happened? The lignification taking place and cells become dead. Here we observed about the structural differentiation, and for it is for the to perform what the hardness to the plant organ. So while coming to sclerenchyma, in sclerids, all the inner walls of the cell become thick walled. And here you can see the neighboring cells may also there, and they are also sclerenchyma. So that is why here you can see the inner and outer wall. Not only it is not like that of inner and outer wall, a cell, the inner walls become highly lignified and thick walled. And here you can see there is no pointed tip is there. No pointed tip is there. So here it is no pointed tip as in the sclerids. So that is the structural difference from sclerids and fibers. Okay, coming to the second one. Here fibers. Fibers you can see what, of course it is with the fibers may show a pointed tip at both the ends, and similarly here the wall of the fiber which is show pit cavity, that is localized unthickened pockets, which is called pit cavity on the cell wall. And here at the center there is a narrow lumen, a small space or lumen is seen in the case of fibers. So sclerenchyma is differentiated into two. One is sclerids and the other is fibers. Sclerids do not show any pointed tip. All the inner walls become similarly or equally thick walled. And as in fibers, it is with a pointed tip and it show a narrow lumen, and the cell walls show the thickening, but some of the pockets which do not show the cell wall thickening or lignification, and those unthickened area which is present in the fibers which are called as pit cavity. So we can conclude, what is sclerenchyma? Sclerenchyma is the kind of tissue consisting of dead cells, and they show lignified or unlignified walls, and it give hardness to the plant organs, and they are of two types. One is sclerids and another one is fibers. Sclerids may show what, all the inner walls become thick walled and it do not show any pointed tip. And as in the case of fibers, it show a narrow lumen with a pointed tip, and the localized thickening become, the inner walls of the fibers may also show the localized, means thickening, lignification or thickening, and there some of the pockets do not show the lignification, and which is called pit. So this is the structural differentiation and the mode of function of sclerenchyma. Okay, thank you.